Thank you. Okay, so we're going to talk a bit about hypothesis testing now. So as I said earlier, this technically, in my view, should come after normalization, but I'm going to treat it first because it's really the essence of what we're trying to do when we do differential expression analysis. Okay, so even before we get to that, actually, I need to add this point about experimental design, is that it's very, very easy to have an experiment that's got confounding factors in it. Okay, so what do I mean by that? Well, if you're going to contrast male versus female, make sure that they're age matched. Okay, so if my experiment has 10 males versus 10 females, but the age range of males is 15 to 25 and of the females is 35 to 45, it may be that the difference you see is sex, but it could also just as easily be age. And so without a balanced design, you just don't know that. Now, the design doesn't have to be perfectly balanced. In other words, you don't have to have an equal number of each type, because the statistical tools are actually fairly insensitive to that. But you clearly don't want things to be completely confounded. So another example may be that if you would say in contrast hearts and livers, you want to make sure that they're from, say, weight matched mice. So otherwise the difference you see might actually just, well, in this case, a heart and a liver is so different that it wouldn't be. But if you did, say, ileum versus colon from normal and obese mice, you might actually not be able to tell which it is, whether it's one or the other. And that goes into the design phase, but you also need to be aware that those effects are actually oftentimes built into an experiment without you realizing it. So other types of technical confounding, so non-biological confounding, which are much, much more common on geo, are things like the date of RNA extraction or the date that the sequencing run was done on it. I know John Story used to teach this fabulous example of an experiment published in Nature, where they claimed that there were 3 different biological profiles of Plasmodium, I think it was. And it turned out that when he downloaded the date of sampling, they perfectly matched the 3 different biological samples that this whole Nature paper was written about. Oops. I've certainly downloaded data sets where I've done my own analysis and said, okay, I'll compare it to somebody else's and not found a match. I remember one example, I was doing a study of craniosynostosis. I had 3 subsets of data. They had 3 subsets, but they didn't match one bit. And I thought, what's going on? And then I noticed in their data set that actually their 3 subsets perfectly matched with date of extraction. When I adjusted for that statistically, my 3 sets matched matched the new 3 sets that I found in their data after adjustment almost perfectly. So that is very, very common in data sets. There is a confounding of date of extraction with some sort of biology. So in that Plasmodium experiment I just mentioned, there probably were biological effects, but they were confounded with the date because they took this phase of the life cycle, this phase of the life cycle just happened to come up at different times of extraction or sampling, whichever it was. Batch effects are less common in RNA-seq. They used to be particularly prominent in microarray studies, so the batch of slides you got. You do need to pay attention to it. For example, 10x Genomics have changed their GEM modules from earlier to a later one because of a lawsuit. There are certainly batch effects in that. You probably don't want to compare a NovaSeq experiment to a NextSeq experiment to a MySeq experiment without doing statistical adjustments. They're equivalent to sort of batch effects. And there's whole samples out there, whole software out there. Combat, I mentioned, COMBAT, is a very, very popular software for adjusting for batch effects. Unfortunately, it's also true that some of us have, some of you have green fingers and some of us don't. I'm one of those people. When I was in the lab doing experiments, I used to have to do things twice usually. But if ever I found a student, an undergraduate who comes in and gets it perfect the first time, I keep them and just don't let them go because they're gold, right? So there are people who are just really, really good at experiments. I've got a colleague at the University of Queensland, Grant Montgomery, whose data is just always beautiful. But in general, particularly in the single cell world, there's massive variability in the quality of the data that you in the QC if you do QC parameters. Other types of technical confounding are things like short end, single end versus paired end. If you have paired end data and you want to compare it to single end data, you might want to just remove the second end and just make it as equivalent to a single end experiment. But even then, there could be confounding. Read lengths, so comparing 35 base pairs to 100 base pairs is going to give different alignment potential, so that will be a confounding factor. And then quality of data, quality of the reads can matter. More often, you're talking about quality of the RNA. So I hope you're all aware of something called the RIN or the RIN, the RNA integrity number. This is a thing called a bioanalyzer trace. So if you take your RNA or your cDNA and you run it out on a gel, it should come out with a certain size range. So you'll get some peaks of the 16S and the different types of ribosomal RNA, and then you should see a large peak between 2 and 5 kilobases, which is the typical size of messenger RNA. If you see a big smear of fragments which are, between 1 kilobase and 3 kilobase, or even say between 500 bases and 3 kilobases in length, then that's a sign that the RNA is degraded. And so this RIN number measures that degree of degradation, and it's a huge confounding factor in all data sets. Now, you can deal with it statistically to some extent, but some journals actually insist that you have a cutoff, or in fact, not so much the journals, as the reviewers. I've certainly had reviewers who've said you need to take out all data that's got a RIN less than 8, for example. So, the better quality of the RNA going in, the better quality of the data you're going to get out. And there is no question whatsoever that there is confounding of these technical factors with biological in almost all data sets. And so when other people reanalyze published data sets, they will often find differences just because of the way they treat technical confounding. Okay, so when we get to hypothesis testing, most of the time, if not all of the time, we're actually dealing with linear models, okay? And there are 2 steps in these, basically. So the first is that you normalize the samples. So back in the days of microarrays, what you were measuring was fluorescence intensities. And the simplest transformation would be to log transform those. We'll see why in a bit. And then you model those as the sample mean plus an array effect. And then what you're left with is residual, and you would do the analysis on the residuals. Okay, so basically what that's doing is saying that if I turn up the laser on the scanner, then I'm going to get much higher read intensities, or if I put twice as much cDNA on the array, I'll get twice as much read intensities. And I need to adjust for that, otherwise I'll find out that all of my transcripts are twice the abundance, okay? So one way to do that is just a simple linear model that fits the array effect and just normalizes things, centers things to the same mean. With RNA-seq data, it's a similar concern. So if I've got one sample read with 25 million counts and another one 50 million counts, then every gene would appear to have twice as many counts as the other. So the simplest thing that people do is to convert their read depth to counts per million. So you basically add up the total number of reads in your sample, you divide through the actual counts by that number and multiply by 1 million, and that gives you counts per million reads. And so your unit of measurement is actually what you would have seen if you had actually read just 1 million reads of each sample, okay? And then as we'll see, there are actually much more aggressive ways of doing normalization. So there are ones that actually don't just want the average read count to be the same, but they want the variance to be similar. I think there are good reasons why that's not a good idea, but it depends on the experiment. And so the main reason why it's not good is because you actually don't expect the variance to be the same across treatments. So if you normalize it to being the same, then actually what you're doing is transforming the data in a way that's not warranted by the data, by the biology. And so there are supervised methods as an alternative for doing that. A problem being that some analysts regard supervised methods as being unreliable, because if you have two different supervised approaches, you're going to get two different results. So which one do you trust? My own perspective is that supervised are better because they maximize your power to see things, so long as you're honest about it. And just applying a simple pipeline exactly the same to all samples is in fact ignoring the underlying reality that there are variants and biological and confounding and other variables that ought to be taken account of. Okay. Nevertheless, once you do your normalization, the next question is to go in gene by gene and assess the significance of treatment effects. So most of the time when we're doing gene expression profiling, we're asking the question, is the average level of expression of gene A different between samples 1 and samples 2? Okay. Now you can also ask the question, is gene A more highly expressed than gene B? That's usually not the target of statistical analysis. That's a qualitative assessment that you can do. Much more interesting in most cases is the question as to whether or not the expression of each gene varies across treatments. Okay. And so we're usually doing this at the level of the log counts per million. And so what we're going to do is to set up models such as, so this is a statistical model is at the expression level. So the counts per million is a function of the mean of the sample, and then all the treatment effects we care about. So sex, for example, genotype, the treatment, drug effect, whatever it is. And we can specify some of these as being main effects, fixed effects, some of them as random effects. So individual within genotype would be a random effect. And then oftentimes as well, we're interested in interactions. Okay, so is there, for example, a sex by genotype interaction? Is there a cell type by COVID status interaction we talked about before? And of course, there's always residual error as well. And we assume that this is normally distributed with a mean of 0. And you can evaluate these models with various types of statistical tests. You can do t-test, it's the simplest. If it's a pairwise contrast, if you're doing multiple comparisons, then you're oftentimes going to do an f-test. So analysis of variance. There's something called a Wold test, which is like a t-test, but not assuming normal distribution of the data. Conceptually, we'll talk a little bit about differences between what are called likelihood ratio tests. So DEC and EDGAR have likelihood ratio tests as one option. And then there's a vast expanding literature on what's the best type of expression test for single cell data, oftentimes indicating that Wilcoxon rank sum tests are best. So basically, if you've got a large number of samples, then it turns out that actually the rank order of genes is itself a pretty good indicator of whether or not there's significant differences. So you have the Wilcoxon rank sum tests ask the question whether the rank is higher or lower in one type of sample than another. So that's different from actually measuring the actual differences in the expression levels themselves. Okay. So, so generally, though, we're interested in asking whether there's a significant difference between 2 or more treatment groups on a gene-by-gene basis. Okay. Now, as with anything, any statistical hypothesis, actually, I'm going to just check the chat here. Yeah. So I'm going to explain this in the next little while, Ron. In any sort of statistical contrast, there are basically 3 things that go into the difference, into the evaluation. So one is the magnitude of difference between the groups. Another one is the variance within the groups. And the third one is the sample size. So I'm just trying to illustrate that here in this toy example. Okay. So maybe my log CPM values or my log gene expression values for a gene in one group are 5, 6, and 4. So, okay, so I've got 3 replicates, and I'm looking at a specific gene, and one of them it's got 4 units of expression. The next one's got 5 units of expression. The other one 6 units of expression. So 4 would be 2 to the 4th, which is 2, 4, 8, 16. So this would be 16, 32, or 64 counts per million in my sample. And then if I look at the other group, where I've also got 3 replicates, I actually see that I've got 7, 5, and 6. Okay. So the mean of these is 5, and the mean of these is 6. So 6 plus or minus 1, 5 plus or minus 1. And so the difference between them is 1 unit. So just saying there's one unit of difference is not really that meaningful. What's meaningful is to say whether that one is large or small relative to the variance within the groups, okay? So just to illustrate that, if I done another experiment where I have a different gene in the platform, and that gene actually has measures of 2, 5, and 8 compared to 3, 6, and 9, then here the difference, the mean is still 5, and the mean is still 6, but this is 5 plus or minus 3. That's 6 plus or minus 3, okay, versus 5 plus or minus 1, 3, 5 plus and 6 plus or minus 1. I think most of you would look at that and say, well, you know, the variance is so large in this second example that that difference is 1 is much less likely to be significant than it is here, okay? So in fact, if you draw the frequency distributions in these cases, they would overlap each other very largely, whereas here they'd be somewhat different, okay? So this will be more significant. The top one will be more significant than the bottom one because that difference of 1 is similar to the variance among samples, whereas here it's very, very small compared to the variance within samples. A sample size effect, you can see here, just intuitively, you can see then that if I actually did double the sample size, now I've got 2 4s, 2 5s, and 2 6s, or 2 5s, 2 6s, and 2 7s. So again, my averages are 5 and 6, but I think most of you would see that even though the variance is the same here and here, the first and the third, I've got twice the sample size, so I'd actually be even more confident in my analysis with this one, okay? So they're the 3 things that matter, is what's the difference between the groups, what's the variance between the group, within the groups, and then what's the sample size I'm looking at. The general modeling framework for linear models is to use something called a general linear model. The alternative is a likelihood approach that we'll get to in a bit, but I like to implement things in the analysis of variance framework that we borrow from agricultural and a whole bunch of quantitative genetics. But the way these general linear models are implemented in EDGE, which I'll do in more detail, are actually not quite the same as the standard classical ANOVA framework, and I'll get into that difference right now. But basically, you're still asking the question whether or not the difference between the groups is large relative to the variance within them, but it's a very, very flexible framework that allows you to evaluate multiple contrasts. So you can have 2 or more effects, you can have 2 or more levels within each one, and you can have interaction effects included in your modeling. So a simple one-way contrast of 2 samples, the mean of 2 samples, you can just get away with a t-test or a simple Wald test. But in this more general linear framework, you can have much more sophisticated and complicated designs. Okay. Now, EDGE mainly uses lemma to perform their one-way analysis variance. As I said, it's a very likely, very powerful framework. But it does usually constrain you to contrast your treatment to a reference sample, or at least the way the software was initially written. It's changed a bit over the years, but a very typical analysis most people do is they're going to specify a reference sample, and they're going to compare their samples to that reference. And we'll see the impact of that in a sec. I encourage my students to use software called LME4, which implements a different general linear model. I just checked this morning, actually. I hadn't looked at the DEC, the EDGE algorithm for some time. They've got their own version of a general linear model now. But I've also come across, some of my students have found this software called GLM, or General Linear Mixed Model, SEEK, which implements LME4. If you just do it yourself, you need to write code to iterate it over all the genes. But GLM SEEK is some sort of complex models, including interaction effects. Okay, and then one other thing we need to discuss is what you're going to get of the output of doing, say, 10,000 different gene expression comparisons is a list of p-values and their differential expression estimates. And so if you've got that, you need to have some sort of significance threshold to evaluate. And we're usually going to do that with a false discovery rate criterion, which could either be a Benjamin Hochberg one or a Q value, and I'll explain that as we go on. Okay. But just what the different types of tests. So a two-sample t-test assumes that the two distributions, so the distribution of gene expression in the two samples is normally distributed. There are reasons why that's not necessarily the case, which is why an extension of this is just a generalization of this is the Wald test. But what you're basically doing is asking whether or not the two population means different from one another, given the standard deviations or the variance within the samples. And so there's the actual t-test computation. And then generalizing that out to S statistics, what we're doing is asking whether the deviations between the samples, we've got a duplication of samples there, between the samples is large relative to deviations within those. So basically, if this is a distribution of gene expression levels among replicates within one treatment group in the orange, and then this is a distribution on the right. Sorry, I forgot to turn on my pointer on again. This is a distribution on the right between the red samples. Then the deviation between the two samples is the mean difference, and we evaluate that mean difference relative to the variation within the samples, which is this is sort of one standard deviation unit there. So an F ratio is really computing the ratio of the between to the within, and given the number of contrasts and degrees of freedom, you can look up charts that tell you what the significance level is. Okay. So generally, you would start by evaluating whether there's variation among all the groups. So in classical analysis variance, the right thing to do is to say, okay, well, here is my 4 groups. So I've got, what was it, B cells and T cells, healthy and COVID, but I've got 4 possible contrasts. To me, the correct way to approach this is to say, are there differences among those 4 groups? And if there are, then go in and do post-hoc contrasts. But more often in gene expression profiling studies, people ignore that step, and they basically will instead go and actually evaluate a specific contrast, such as are the dark blue different from the light blue, or are the dark blue different from all of the rest? Or in a reference sample sort of setting, what you're doing is saying, are the dark blue different from the light blue? Are the pink different from the light blue, which is our reference? Are the red different from our light blue? And so you might find that all 3 are different from those, and you're going to get a different result than if you say that my reference is the red, because pink versus red is not going to be different, whereas these 2 may be. Okay, so specifying your reference does matter, but in classical ANOVA, you don't do that. You just start out by asking are there differences among the groups. And then this difference between type 1 and type 3 sums of squares has to do whether or not you're including random effects in the model. So whether or not you specify, say, the difference between blue and dark and light blue is random or fixed will impact the sums of squares that go into this computation. And then, of course, the other level of analysis is whether or not you're incorporating random effects of individuals within samples into the analysis. And tomorrow, I'll actually show a case in single cell data where that really matters. Okay, so this is something called a volcano plot that we actually first published with Russ Wolfinger about 20 years ago now. And this data set we analyzed was a Drosophila data set we had, where we were asking whether or not there was an aging effect among strains and sexes. And so this is just a volcano plot showing differences between male and female whole body flies. And so what you're seeing here is each point is one gene, and you're representing the fold difference between gene expression between males on the right and females on the left, okay? And on this particular scale of a log base 2 counts, 1 is 2-fold difference, 2 is 4-fold difference, and 3 is 8-fold difference. So a difference of 1 on the log 2 scale corresponds to a 2-fold change, okay? And that's in both directions. So -1 is a half, -2 is a quarter, -3 is an eighth. And so a 4-fold difference, so a 2-fold difference between -1 and 0 is the same as a 2-fold difference between 0 and 1. It's just symmetric, okay? And so you can see that there's a range of differences from almost no difference up to very large differences. Now, in most data sets, you'll see a huge number of points clustered around 0. In this male-female fly comparison, most genes are differentially expressed, so you don't see a big cluster down there, but that's somewhat unusual. Now, you should be asking yourself, what's the y-axis? So in this case, in a volcano plot, what you're plotting is the significance as as a negative log on the base 10 scale now of the p-value. Okay, and the reason we do that is that when you take the negative log of the p-value, then very small p-values correspond to large numbers. So if I just plotted the p-value itself, then these highly significant things would just merge into the x-axis. So by taking the negative log 10, we spread it out. Those of you who are used to GWAS, it's the same thing. In a Manhattan plot, you plot the negative log p-value on the y-axis. And so you get this spread. And what you observe is that there's a general correlation between fold change on the x-axis and significance on the y-axis. And what you get with a volcano plot is you see that there's this bidirectional effect. In fact, when Russ and I first did it, we actually plotted the negative log p-value of the negative ones below the axis and had what we call a butterfly wing plot, but it would take up too much room. So instead we made it into these volcano plots. And in any case, what you get then is 6 sectors or 4 types of sector in the plot that are informative, okay? So in the middle here, the red ones, panel D, region D, the genes which are not changing much. So the fold difference is less than 2-fold, and the significance is less than some cutoff here of 5, okay? And so that's plotted there in a minute, okay? So D, we can all agree there's not much change and it's not very significant. So we'll call those genes the non-differentially expressed ones. And then conversely, in regions A, either up or down, so higher in males or higher in females, or low in males, are ones which have a big fold change, so more than 2, and they're highly significant, so p-value less than 10 to the minus 5, okay? So we can all agree that these are differentially expressed genes up here in the green quadrant. And then you get these 2 other quadrants. So quadrant C are ones where the fold change appears to be large, but they're not very highly significant. Okay, so depending on whether or not you care about fold change or significance, you're going to include these. So prior to about 1995, most papers really just asked you to find genes that were 2-fold or more. Quickly, the field has transitioned to realizing that significance matters as well. Okay, so of course, once the statisticians got involved. Panel B is highlighting why that matters. So these are genes where it actually looks like they're highly significant, so p-value is less than 10 to the minus 5, even though the fold change is nearly not that much, not that great. Now, there's nothing magical about 2-fold, of course. Okay, so a 10% difference in expression of a kinase, for example, or a transcription factor could be biologically very, very meaningful. It could have a big consequence, whereas a 10% difference in a maybe actin or some enzyme maybe doesn't make that much of a difference. So there's no real rules about that. But the important point is to notice that there's nothing magical about 2-fold. And you can ask the question, well, why do people use 2-fold? I think the answer to that is actually 2 is just an easy number, lazily. But also it actually turns out that something like 5% of genes in typical experiments are differentially expressed. So just intuitively 2-fold made some sense, but there's really nothing magical about it. The other thing you need to contemplate is that if you're doing bulk RNA-seq, you've usually got a mixture of cell types. And so if you have like a 5-fold difference in a cell type that's only 20% of the cells, but no change in the other 80% of the cells, then across all of the cells in that bulk sample, the difference will just be 2-fold. Okay, so if you have a minor cell type with a large change and you're using a 2-fold cutoff, then actually you would expect many of the genes to be less than 2-fold. So they'd be in this quadrant B or D, and you're not powered to see them just because they're in a minor cell type, and yet they're nevertheless very important. So if you grind up total brain, for example, maybe there's a neuron that you care about, but you're never going to see it because you haven't isolated that cell type. Or similarly in blood, if you're looking at, say, macrophages, which are a minor cell type, there could be a large change in them, but you'll miss it because they're actually submerged by the total bulk RNA-seq data. All right, so a few other things about this plot. So why did I color at 10 to the minus 5? Okay, so that goes to our multiple comparison issue, okay? So if I've got, say, 5,000 genes in an experiment, I don't expect any of them to be significant less than 10 to the minus 5 at all. I don't expect to see a fold change that much. Typically, what people do in multiple comparison adjustment is something called the Bonferroni correction. So you take the number of tests that you've done and you take your nominal p-value. So normally that nominal p-value is 0.05, okay, or 1 in 20. So if you're just testing one thing at a time, then you typically will say, if I'm less likely to see that in 20 replications of my data set, then if I see it 1 in 20 times, then that's rare, and I'll accept that as a significant result. But if I'm now evaluating 10,000 genes, I expect to see 1 in 20 of those at that 0.05 cutoff, which would be, out of 10,000, that would be 500 genes significant at 0.05. So on this scale, 0.05 is about 1.3 on the negative log 10. And so that would be a cutoff across here. But it's many more genes, but it's hundreds of genes. And so I don't want to include them, right? So I can have different criteria. So one is to do what's called a Benjamini-Hochberg comparison or false discovery rate control. And that is, when I'm doing that, what I'm doing is saying I'm going to control the false positive rate. So I'm going to say I'm willing to do an experiment where if I have, say, if I expect a cutoff of where I expect to see 100 genes differentially expressed and I see 1,000 genes differentially expressed at that cutoff, then I know that only 10% of them are false positives, that 900 out of those 1,000 are true positives. And I may not know which of those 1,000 of the 900, but I'm much better off including 900 genes in my, or 1,000 genes in my downstream analysis, accepting that some of them are false positives, than I am of just taking 100 genes, which I'm much more confident about, okay? So alternatively, we do much more strict thing, which would be a Bonferroni cutoff. So 0.05 divided by 10,000 genes would be would actually be 10 to the minus 5, right? So no, say 5,000 genes, 0.05 divided by 5,000 would be 5 by 10 to the minus 2 divided by 10 to the minus 4 would be 10 to the minus 6. That would be a cutoff there. But that's usually too strict. So you'd find that there's too many genes. You won't get enough genes. So a Bonferroni correction, even though that's what's used in GWAS, is typically not used in gene expression studies because what we care about is being more inclusive. So there's just, in the field of gene expression studies, we're better off doing downstream analysis with a panel of genes where there's some false positives, whereas in GWAS, we need to be confident about every single gene. At least that's the way the field evolved. Okay, so a typical cutoff is set at a level where we're controlling the false positive rate. And I've just set it here as being 10 to the minus 5, as being the one that gives me this optimal control of that. And then the final thing I wanted to sort of mention in this particular plot is you should be asking yourselves, well, what's different between this gene here, which is twofold, but significant at 10 to the minus 10, and this gene here, which is twofold, but not even nominally significant at 0.05. Okay, well, what's different is extremely high. So in my 3 replicates, I probably saw very close to the same measurement. Whereas in this one, I might have seen one sample that had a 10-fold difference and another 4 that had no difference whatsoever. So the average fold change was 2-fold, but it had high, because of that high variability, I got a small p-value, okay? And again, it gets to this idea that what you're evaluating is not the fold change itself, but the fold change relative to the variance. So the reason why you get a volcano plot like this is that you're actually seeing the more significant genes, ones at a certain cutoff, are the ones that had more repeatability to them. And so they're more reliable estimators. And if you follow that reasoning through, then it actually implies that actually many of these genes in here that have a relatively small fold change, but are nevertheless highly significant because that small fold change was repeatable. So if I see a 1.2-fold change time after time after time, then I actually believe that it's probably significant. Just kind of check. So hopefully I'm answering some of these questions. Jorge, no, absolutely not mandatory to use a reference sample design. Less than common. I think actually we might come back to some of these questions at the end of the day. I'll go through the lecture and then we'll come back to some of these because I might answer some of them in the intro. Okay, so there's actually another way of drawing a volcano plot. So Gary Churchill's group, around about the same time as we published our study, published this plot here. So actually 10 to the minus 5 is a conservative Bonferroni correction for 5,000 genes. So it's 0.05 divided by 5,000 is 10 to the minus 5. I think I misstated that earlier. I in my head calculated incorrectly. So that's the conservative Bonferroni correction. A more typical false discovery rate correction is going to be something like, typically it's around about 10 to the minus 3, depending on the magnitude of the experiment, sample size, and things like that. But what I wanted to say now, though, is a significant testing is actually, so it's allowing you to include genes in sector B. But what Gary pointed out was that actually there is a lot of variance to your denominator term in these analyses, okay? So that all genes don't have the same variance. And that estimate of the variance itself is subject to sampling bias. So he employs something called a shrinkage estimator. So sort of you sort of do assume that genes with a similar fold change have a similar variance. And when you do that, you sort of shrink down the denominator variance. You're using a more common denominator variance. And if you do that, then it actually implies that actually you expect that Russell's represented in their job because they actually do a very, very strong shrinkage estimation adjustment for this fact that there are differences in the estimation of the variance from experiment to experiment. Okay, so in a volcano plot like this, we're not taking into account that there is actually error in the variance estimation. If you do that, you're more likely to get a result like this. Okay, all right.
Hello, everyone. Looks like Greg has some internet problem. Hopefully he will be back in a few minutes or less than that. Can you guys at least hear me? Okay, all right. Let's give Greg a little bit of time. I'm sure he will be back here as soon as he can. So just now, Greg was about to talk about false discovery rates and Q value, and that's sort of in general related to I would say correction for multiple hypothesis testing. So I guess one question maybe for you guys in terms of your preparation, have you guys heard of what it is and try to do some of it? Yes? No? All right. So well, I'm sort of expecting the answer. Usually when you do a hypothesis testing, for example, t-test, right? And you will get a test statistics, which is roughly, let's say you have two sets of numbers. One set of number coming from measurement of a gene in a under a particular condition, let's say control condition. You have 10 control samples. You have measurement for one gene for these 10 control samples, so you have 10 numbers for your control condition. Let's say you also have 10 numbers for a case condition that you're interested in, right? And you have 20 numbers, 10 versus 10. And you can say that, hey, we can do a t-test to ask whether the first 10 numbers and the second 10 numbers are significantly different from each other, right? And then running t-test or any other statistical test, you will basically get two things. One is a test statistics, and the other is a p-value, right? The test statistics usually, for example, for t-test, is the difference between the mean over the overall variance. But there are also other test statistics you can do. And then there is associated p-value that's usually between 0 and 1. 0 is good, 1 is really bad. And now one thing that I think Greg was about to show, okay, he's here. So if, let's say your data is totally random, you get thousands or millions of randomly simulated data sets and you run t-tests for each of them, right? And then by chance, you're gonna see some good p-values and some bad p-values. And if you look at all the p-value from all the random data, you get a uniform distribution, okay? Which is basically the distribution in the bottom left corner, but without a spike all the way to the left. And whenever you do many, many tests, if there's nothing interesting there, you see a uniform distribution. But if there are some interesting things there, you see the spike all the way toward the left. And then depending on how much higher that spike is compared to the rest, the flat, okay, that gives you roughly like how many real signals are there among the millions of tests you do. And that's sort of one way to interpret FDR and Q value. All right, so Greg is back. And I'll stop hijacking the conversation and let Greg continue. Greg, you're still on mute, and we don't see you. But we see your mouse moving. Okay, we see you now.
Okay.
And
I'm sorry about that. I have no idea what happened. The internet went out even though my signal was strong, and then it just came back on. So I have no idea. So I apologize about that. The perils of teaching online. So I'm just going to go back to the PowerPoint. So yeah, so as Peng just said, and so it's nice to know that you can take over if this happens again, Peng. On a uniform distribution, the expectation is you have the same number of p-values in each bin, but actually there's so much covariance of gene expression, and there's so many significant results in most experiments that you expect to see an excess of p-values less than 0.05 in this case. So each bin is 0.05, 0.1, 0.15, 0.2, and so forth. So these are sort of 5% bins of p-values. So what this is showing you is that there's an excess there. And you can actually use that to actually estimate the true fraction of nulls in the experiment. And that's sort of where asymptotes. So it's across here. So in this particular case, the true number of nulls might be around about 70%. So there is a little bit of signal here, and then there's a lot of signal here. In fact, it may be that none of these genes here actually pass the strict Bonferroni threshold, but nevertheless, there's signal there. So the Q-value procedure was used, and it's not typically seen so much anymore because single-cell RNA-seq is so powerful. But nevertheless, so there's a monotonic relationship between p-value and q-value. This lambda is just your estimate of what the true null percentage is. But if you do this and you adjust your expectation, so your false discovery rate criterion to the expected number of nulls, then you can actually include more genes. So it is conservative, but it's statistically conservative, but it's also allows you to include more genes in your analysis, okay? So that's a little bit different from just a simple Benjamini-Hochberg adjustment, which sort of says, okay, so in 10,000 tests, I expect 100 of them to be less than 0.01. And if I observe 1,000, then my false discovery rate is 100 divided by 1,000. If I used a Q-value procedure, then I actually may be able to reduce that, maybe actually go out to 0.001 and see genes at that level. Or I might be able to go down to 0.03 and still have a 10% rate. Okay. So if you're doing bulk RNA-seq, I certainly recommend the Q-value procedure, and that's easily implemented in bioconductor. All right, so this is just a way of sort of thinking about how we do hypothesis testing. How to get rid of that? Okay. All right. So types of contrast that we can do. So let's suppose that these are just 5 samples, and so I've got to imagine a y-axis is gene expression level, and I've just grouped 6 replicates of each of 1, 2, 3, so 6 samples here. So classical analysis of variance, what you're doing is just asking, are there differences in the mean value among those 6 groups? And if there are, then I go in and do post hoc contrast. So I might look at it and say, well, I think the red ones or the red and the blue at the top there are different from the others. And so your first step is just to ask if there's variation among groups, given the variation within the groups. Usually, though, what we're doing in gene expression experiments is actually evaluating a very specific contrast. So you're saying, for example, in blue there are the female samples, which is the top one, the top blue box, different as a rule from the male ones. And you can actually, simplest way to do that is just to ignore the fact that they're from low, moderate, and high treatment groups and just treat them all as one. And so you compare all of those top 18 samples against the bottom 18 samples. But I think you can imagine that actually a better test is to evaluate those 18 relative to these 18, given that you already know and incorporate the variation within the groups, okay? And then there's 2 ways of doing that. It's either treating that as a random effect or a fixed effect, but I don't want to go into that now. Alternatively, you may ask the question as the differences between the low group and the moderate group, or the low group and the high group, or the moderate and the high, or in general, in the ANOVA framework between low, moderate, and high. Okay, so that's sort of how we go about modeling. So where you're specifying your contrast of interest in advance. Another thing you might do is actually go in and actually say, well, I actually want to know specifically whether or not, for example, high treatment males are different from low treatment and moderate treatment females. So I can actually, in that sense, essentially ignore the other 3 and just do a comparison of these ones to those ones. And the way you do that in EdgeR or DE-seq is actually to specify these ones as 0 or null in your model, and then these ones to say -1 and that one is +1 in your experimental design matrix, and allows you to do those very specific post hoc contrasts. If I'm talking about an interaction effect, what I'm getting at there is asking the question of not just whether males and females are different, and not just whether low tend to be different from moderate from high treatment groups, but whether or not there's interaction effect. So an interaction effect would be, in classical quantitative genetics, crossing of line means. Okay, so if you extend these out, then the regression through the treatment effect of the females would eventually cross the regression to the treatment effect in males. So in this particular case, there is no treatment effect in the males, but there is a treatment effect in the females. And so an interaction effect would actually statistically tell you that the slopes of these 2 lines are different from each other, okay? And actually, you can evaluate the significance of that. You could imagine that those two slopes are different, but not significantly different. So in a very powerful ANOVA framework, you can actually evaluate that hypothesis. So in this case, you'd probably conclude that given the variability within each of the treatment groups within sexes, that actually there is a difference where the high treatment reduces gene expression in females, but has no effect on gene expression in males. Okay. So there are actually different ways of evaluating those sorts of contrasts. So one is a t-test. And so we've already talked about that. It's assuming that the data is normally distributed. That's oftentimes not the case, particularly for low abundance transcripts where they maybe have a large number of zeros. So the data is what we call negative binomially distributed. And in that case, you more commonly will see a generalization of the t-test to what's called a Wald test. This is the ANOVA framework. So the F ratio is evaluating the variance between treatments to within. We've discussed that. If many of you are maybe familiar with EDGAR or DE-seq fitting a likelihood ratio test. So what you're doing there is evaluating whether inclusion of a parameter in a model gives you a better fit to the data. So reduces the variance, the unexplained variance. And so the likelihood ratio test statistic is asymptotically chi-squared distributed. And so you take -2 times the logarithm of the actual ratio of the likelihood of the data with the parameter, without. So what you have to do is to actually just take your baseline model, which is the null hypothesis basically, and then you compare that to a model where you actually fit a fixed effect of treatment or a fixed effect of age or an interaction effect. And so that's a very powerful framework. I just noticed this morning that EDGAR actually really does recommend their implementation of the general linear model analysis of variance. So they've got a modified F ratio in their analysis. It's adjusting for this, essentially doing their shrinkage of the variance estimates. And then another thing that you'll see, particularly in the single cell data types, is a rank sum test, which is Wilcoxon signed rank test for paired samples, equivalent to a Mann-Whitney U-test. And the beauty of that is you're not actually assuming anything about the distributions. So you're testing for symmetry of the ranks about the mean without actually comparing the actual magnitude of the deviations. And it turns out that that's actually a fairly strong test in the single cell framework. And you also see it implemented in ATAC-seq data. We'll see in a couple of days' time and other types of data sets. It may actually be superior. But it's very, very dependent upon the sample size, upon different aspects of the data. There are vast literatures out there comparing the performance of these different testing programs. And the bottom line is that different ones perform differently under different conditions. We know a little bit about that. But basically, analysts have their preferences. You just need to be aware that the way you model things will impact the outcome, in some cases, quite significantly. Okay, so I'm not going to go DE-seq2. So the two major tools that people use are DE-seq or DE-seq2 these days, and EDGAR. EDGAR is out of a group from the University of Melbourne, so I'm biased to sort of reporting on it, even though I'm a Sydney-sider. But EDGAR offers this sort of basic workflow. So you basically read in the data, you specify an object you're going to do the gene expression analysis on. You specify the factors of interest or the groups. So you make a vector of factors. So those factors would be sex, genotype, treatment, whatever they are. And then you specify whether or not to include them in the model. You can do a filtering step. So you may say, I only want to include genes that are expressed in at least, say, 5 out of the 50 samples, or I may only want to include genes whose log fold change is greater than this or that. So there's a step there, so you can keep those. There's a normalization step. So this is, I won't go into this now, but it's going to calculate a factor that normalizes the data appropriately. So adjusting for outliers is one thing it does. We'll talk a little bit about that. And then you specify your model, and then you estimate the dispersion parameters. So whether or not how much the variance is within and among groups and treatments. And then it's giving you two options. So this quasi-likelihood F-test, which is likely analysis of variance, is the one that they recommend. I think it says up here, the quasi-likelihood method is highly recommended for differential expression analysis of bulk RNA-seq data. That they didn't have that a few years back, but this is what they're recommending now. Whereas the likelihood ratio test can be useful in some special cases, such as single-cell RNA-seq, and or if you have data sets without replicates, is the recommendation there. Okay, so our preference is actually quasi-likelihood F tests. They're much more conservative than the likelihood ratio test under some circumstances, but you need to read the code to find out how to do it. As I mentioned earlier, if you go down further in their user's guide, it gets very, very statistically sophisticated very quickly. So if you want to understand it, you need to take classes in statistics or talk to a statistician. But nevertheless, the vignettes are sufficient to allow you to start implementing those models fairly quickly. All right. So how does hypothesis working testing work in EdgeR? So if you compare the output of likelihood ratio tests in EdgeR to those of a classical t-test or f-test. So I put this slide together a few years ago before they were offering these their modified f-test. Particularly when you look at small n's, you're going to get very, very different results. Okay. So I used to get very frustrated because my students would be running things in EdgeR and I'd be running them in SAS and be seeing very different results. Turns out that if you get large sample sizes, they converge on each other. But there are, for small samples, all of these tests are going to give different results. And there are a bunch of reasons for that, among which are the following. Okay, so an important one is, and I think this is a good thing in EdgeR, is it does something called TMM normalization, or trim mean of the median, or median of the mean. So what it's doing is it's adjusting for the fact that in your library, once you've got the counts per million, there could be a few high abundance transcripts that are really throwing off the normalization. So if you've got hemoglobin in blood, or if you've got actin in muscle, or if you've got rubisco in plants, then as much as 50% of the reads could be due to just 1 or 2 or 3 genes, or as many as 20 genes. But you don't want them biasing the analysis. Ribosomal genes in RNA could be a very high proportion of the counts. So what TMM is doing is saying, okay, if those few genes are 50% of the reads in sample A and 30% in sample B, then actually it means proportionately that there'll be higher average expression in sample A than average sample B because they're accounting for more of the reads. So TMM normalization is adjusting to that sort of bias. DESeq has a similar adjustment. The second thing that's happening is that EdgeR is shrinking this variance of low abundance transcripts by fitting something called the negative binomial expectation. So it's actually doing its t-test, doing its statistical tests, not assuming a normal distribution, but rather assuming a negative binomial distribution of the data. And I'll show you tomorrow what that looks like. But conceptually, it's not really doing this, but basically think about that if you've got a low abundance gene, and you've only got, say, 20 million reads, there's a reasonable likelihood that that gene will actually not appear in the data set. So negatively binomial distributions were developed to take into account the fact that there's a likelihood of failure or a zero count in this context every time you do a trial. Okay, so if you were to repeat the experiment time after time after time, you'd actually get a much better estimate of the true abundance than just doing it a single time. And so the actual data you get from a single RNA-seq run, it turns out, is at the low end negatively binomially distributed. So there are genes where you get null counts, but that null doesn't really mean it's not expressed. It actually means it just didn't appear. So when you do your testing, you're taking into account that by fitting this negative binomial model. Okay. And then particularly in this adjusted F ratio test now, what EdgeR is doing is actually performing a very powerful Bayesian within sample variance adjustment in its general linear model fitting. And actually, I think that's where most of the gain in power comes from, in my view, is in this step here, rather than in the negative binomial, because actually at the low end of the scale, you actually don't have much power anyway to see differences. So if you actually look at the significant genes, they tend to be genes that are outside the range of those low abundance ones. Okay, but this when I showed you Gary Churchill's plot, I made the point that the genes with a similar expression difference nevertheless have different variances, and you adjust for that. So that's where I think there's a lot of gains in the way that the Agile model codes its general linear model fitting. So he now implements this quasi-likelihood F-test to do that, which should give you similar results to analysis variance. Okay. Now, for more complicated analyses involving two or more factors, which can include nesting, so individuals within a treatment group, they can include random effects, so individuals usually random. The modeling effect frameworks are quite different. And so the adjustment for the different dispersions really matters a whole lot. So I actually historically have preferred running prop GLM in SAS, although it doesn't make this shrinkage estimate adjustment. So Jump Genomics actually added, some of you may have access to that. It's actually about to be phased out, replaced with Jump Plus, but nevertheless, SAS is industry leading standard. The equivalent in R is LME4, or implemented now in something like GLMM Seek. Okay, so anyway, they're different modeling frameworks. So how do you handle this? So let's go back to our toy example. In this 2x2 design, there are two cell types, so the dark blue and the dark red, and two treatments, so the healthy controls and the COVID. So I'm sorry, the cell types with a light and dark in each. And so we had three individuals with duplicates of each sample. So these likelihood ratio tests allow you to model specific contrast under the assumption that the effect of interest is present relative to a simple model without any effect. And if the effect is significant, then more of the variance is going to be explained by the model than you expect, given the dispersion in the data set. Okay, but as I mentioned, it matters as to what you specify as a reference. Okay, so we can set up different models. I can ask, are the pink and the red, so are the COVID samples different from the light blue and the dark blue? So is there a COVID effect? So the simplest model is just to ignore the cell type difference and just do that contrast. I could maybe also ask, are dark blue and dark red different from light blue and light red? So in other words, is there an overall COVID effect? Okay, so that's a different model. Or I could ask, are the dark red different from all of the others? Okay, so in other words, are the B cells in COVID unlike any of the other cells? Okay. So what you're going to learn from the vignettes is how to fit those models. Okay, and so basically you're assigning weights. So if you want to ignore some of the samples, you give them a 0, but otherwise you have to get the others to sum to 1 or -1, and then you can specify whatever contrast you wish. If you're going to fit more complex models with random effects, nesting and interactions and so forth, it actually requires special skill to get that right in that data there. But so that's where you really want to start talking to an expert statistician to help you to fit the models most appropriately. Okay. So I'm just going to finish up today with a few thoughts about power. So I stole this figure from the internet because it illustrates the notion of power, but it's actually not a generic, it's just a generic plot. It's not representative of RNA-seq data. But what you're doing in a power analysis is satisfying grant reviewers who want to know how likely you are to see a fold change of X for a given sample size at a defined probability level. Okay, so the red curve might be a small fold change of 0.1, the green curve, a slightly larger fold change of 0.15, and the blue curve, an even larger fold change of 0.2. So the larger the fold change, the more power you ought to have. So what you're doing here is saying, I've got, say, 90% power, so 90% likelihood of seeing this twofold change with a sample size of 80. Whereas if I want to see the 0.1 fold difference, then I'd actually need a sample size of 300. So that's typically the question that reviewers are asking you to put into a grant proposal, is how likely am I to see fold change of gene X for a given sample size. However, the fact that genes have different expression levels means that the fold change itself isn't really what you're interested in. What you're really asking is what's your power to see a difference in the proportion of the variance explained by the effect in your model. Okay, so instead of differences here, what you should be thinking about is the proportion of the variance explained. And these two papers I've added at the bottom here, so there's 2017 one and a 2014 one, two pretty good discussions of how you do power analysis in RNA-seq studies. And there's software that they'll point you to for doing these calculations. And they're adjusting for dispersion parameters, the negative dynamic distributions, and so forth. So it's not a straightforward thing at all, but there are power calculators. Personally, I think it's a somewhat pointless exercise because there's just enormous variability in what fold changes are biologically meaningful. There's nothing magical about, twofold or 20% of the variance explained or 10% of the variance explained. I think we're much more interested, in my view, is asking what are the relative effects of different components in the model. So is sex more important than age, more important than drug treatment? However, the fact is that reviewers typically do want you to specify what your power is. And the good news there is that if you do these power calculators, very often it turns out that if you have 4 replicates, or so say 4 viable biological replicates of each of your treatment groups, as a rule of thumb, that's usually enough to give you very good power to find hundreds of genes. But the variance among modeling methods is usually larger than the variance due to sample sizes anyway. So I do it. I put power calculations in my grant proposals, but I always do it sort of holding my tongue because I really don't see the point of it. The fact is that the larger the sample size, the more power you get. The other final point I'd say on that is that you can get power in a small experiment with 3 or 4 of each. But without replicating that experiment completely independently, preferably from a different time, maybe in a different lab, you're never going to be sure that that difference you see wasn't actually due to some confounding factor you haven't even considered. Maybe a batch effect or a date effect or some environmental parameter. So larger experiments are always better, not just because they increase power, but also because they tend to control confounding factors that you may not have thought about. Okay. So I'm going a little bit over for the day, but that's good because what we like to do is to finish the day by giving you a chance to ask questions as a group before we retire for the night. So I've got one. Okay, so if I'm doing 10,000 tests,